overall goal of this cell microarray platform is to correlate measurements of both cell differentiation and traction forces as a function of microenvironmental context.
This method can help answer key questions in the field of tissue engineering, enabling both fundamental investigations of stem cell biology and optimization of differentiation protocols. The main advantages of this technique include its throughput, ability to vary biochemical and biophysical cues, and endpoint readouts by both immunofluorescence and traction force microscopy, or TFM.
Though we have used this method towards understanding liver progenitor differentiation, it can be readily applied to other adherent cell types and tissue contexts.
Visual demonstration of this method is critical, as experimental success depends on integration of the arraying protocol with both high-quality hydrogel substrates and traction force microscopy.
To fabricate fluorescent bead-containing polyacrylamide hydrogels on a silanized 35mm glass bottom petri dish for live evaluation of cell-substrate interactions using TFM, first, prepare glass substrates in solutions as described in the text protocol. Place silanized 35mm glass bottom petri dishes into a glass drying tray and pipette 20 microliters of 9:1 pre-polymer bead to photoinitiator solution onto the center of each dish. Gently cover each dish with a 12mm circular cover slip while avoiding the creation of bubbles.